Okay. So basically okay, tak apa. Kalau yang tak dapat tu tak apa. Okay. Saya nak tanya you all dah habis kan belajar chapter macromolecule?
Dah.
Yeah, right. Okay, so basically for our tutorial today, okay, we are going to go through, okay, together the question for part A, MCQ question. So last time we have done up till number 15, okay, 15. So for today tutorial session, we are going to continue to look at question 16 and so on sampai habis lah kot yang soalan objektif ni, alright? So okay, so we go through the question. Okay, it says here, sample W which contains carbohydrate is added into test tube 1, 2 and 3. Okay, so so test tube 1, 2 and 3 contains substance, contains carbohydrate. So you have to determine which group. Okay, so for test tube 1, the treatment is tested with Benedict reagent, so no color change. So basically Benedict reagent, as you know, it is you use the reagent in food test to detect the presence of reducing sugar kan. So reducing sugar, you have glucose, okay, galactose, fructose, okay, all the monomers, and then the two disaccharide that it can detect is maltose and also
Lactose.
lactose. Okay, so obviously kalau yang okay, no change of color. And then kita tengok number 2, warm with acid, neutralized and then tested with Benedict reagent. So it produces brick red precipitate. Okay. Okay, so this one it should contain reducing sugar. Okay, yang ni tak ada reducing sugar. Yang number 1 does not contain reducing sugar. This one contains reducing sugar because it tested positive. And then tested with amylase. Okay, amylase solution and then tested with reagent and then it produces brick red precipitate. Okay, so as you know, amylase is the enzyme that breaks down starch. Okay, starch. When you eat food, okay, when you eat your rice, for example, bread, okay, so the amylase that contains in your saliva will break down the starch, okay, into a smaller molecules. Okay, it can be disaccharide, it can be monosaccharides. Okay, so bila dah when you have mixed, okay, your food sample with amylase and then mixed with a Benedict reagent, it gives you a positive test. So it means that your starch has now been broken down by the amylase into monosaccharides. Okay, so which carbohydrate are present in sample W? Okay, so kalau sample W, obviously bukan maltose lah kan, bukan glucose. Okay, so so the sample contains sucrose and also starch. Faham So the answer would be B lah. Ada yang tak faham?
Faham.
Okay, so yang ni nanti recording ni saya akan bagilah and then you can go through yang mana yang tak dapat buku lagi. Okay, so number 7, which of the following polymers can be formed from the monomers shown in figure 4. Okay, so as you can see in figure 4, it shows you the structure of what?
Glucose.
You have 2 types of glucose, right? Which is
Alpha.
And
Beta.
so in the figure it shows you which one? Is it alpha glucose or beta glucose?
Beta.
Beta. So beta.
Beta.
Alright. So beta glucose. So obviously the monomer for amylose is alpha glucose, amylopectin alpha glucose, starch is also alpha glucose. So the answer will be only cellulose. So the answer is A. Kan? So how do you know it is beta glucose? You look at carbon number 1 and the hydroxyl group is above the plane, okay, of carbon number 1. Okay, so kalau alpha glucose OH dekat bawah, right, of carbon number 1. So you have to know how to number the carbon. This is carbon number 1. You have oxygen, okay, and then carbon number 1, carbon number 2, number 3, number 4, number 5, and number 6. Okay, so you have to know how to number the carbon for the glucose, especially glucose, so that later you know how to name the bond. Sebab apa? Sebab dalam carbohydrate you have to know that there is 3 types of bonds. Okay, so you have 1,4 glycosidic bond, you have 1,2 glycosidic bond, you have 1,6 glycosidic bond. So you have to know where, okay, will these glycosidic linkages present. Okay, so kalau dalam structure maltose it is 1,4. Okay, 1,4 glycosidic linkage. Kalau dalam structure starch, okay, it can be the monomers are linked by 1,4, but when you have branches, starch have branch kan. Kalau which one have branch? Is it amylose or amylopectin? Ini adalah dalam starch kan, component in starch. So starch ada 2 jenis. If it has branch, so it will be, is it amylose or amylopectin?
Amylopectin.
Amylopectin kan. So the branches are hold together by 1,6 glycosidic linkage. Okay. Tapi kalau 1,2 glycosidic linkage, it occurs between what? Between the monomers in the structure of sucrose. Okay, so saya remind, tolong tengok balik awak punya lecture note. So tolong tengok balik video so that you remember. Soalan yang macam ni dia selalu keluar untuk awak, untuk test your understanding on how to name the bond. Okay, you have to know how to name the bond. You have to know how to draw. Okay, how to draw the structure of maltose. How to draw the molecule of sucrose. So you have to know how to draw the structure of lactose, okay, between the monomers. So kalau dalam sucrose, the bond is 1,2 because it involves the bond to form between fructose and also glucose. Okay, so kalau fructose dia punya cara number dia special sikit kan. So dia bentuk macam sekejap. Ini 1, 2, 3, 4. Macam ni kan. Okay. CH2OH kan. CH2OH. Kalau tengok kat sini, yang ada yang kat atas ni carbon number 6, tapi kalau yang fructose, you start with here. Okay, 1, 2, 3, 4, 5, 6. Okay, so that one you have to know how to number. So kalau dalam structure sucrose, it involves, this one is fructose and the other one is glucose here. Okay, so the bond will be formed between carbon number 1 and carbon number 2. So that's why we call it as 1,2 glycosidic linkage that is present in the structure of sucrose. So you have to know where are the bond present. So ingatnya kalau 1,6 dalam structure glycogen pun occur, glycogen kan. It also occurs in glycogen and it also occurs in the structure of amylopectin. Amylose tak ada. So amylose sebab apa? Amylose does not have branch. The structure of amylose is just helical like that. Okay, tapi kalau amylopectin, it is helical, okay, and then you have branch. So where you have branch is 1,6. Kalau glycogen, you have more branch. Kan, you have more branch. The structure of glycogen is similar to amylopectin, except that it is more complex because it has more branches. Okay. Okay, so number 18. Okay. It says here starch and cellulose are cis transisomers of each other, polymers of glucose, used for energy storage in plant, structural component of plant cell wall. So salah lah yang C and D. So C and D, this one is for starch only. The statement refers to starch only. Okay, kalau D, it refers to cellulose. Okay, kalau yang E ni, cis transisomers has nothing to do with these two structures. And then obviously the answer will be B, polymers of glucose. Okay. Kalau saya nak tanya, kalau dalam cellulose, okay, can you name the bond that hold the beta glucose together? Glucose, the monomer is beta. Cellulose, the monomer is beta glucose. Okay, so what are the bonds that are present, okay, in the structure of cellulose that holds the monomer together, the beta glucose?
Beta 1,4 glycosidic bond.
Okay, so beta 1,4 glycosidic bond. Sebab apa? Dia melibatkan beta glucose. Kalau yang ni pun boleh letak alpha kalau macam dekat dekat maltose kan, because it involves it involves alpha glucose. So to be specific, the name is alpha 1,4 glycosidic linkage. Okay, but if you are not sure, during the exam, sama ada dia alpha ke beta ke, so you can omit the alpha and also the beta in the name. Tapi mesti kena ada yang ni 1,4, 1,2, 1,6 tu. Okay. Okay, number 19. Choose the correct pair of carbohydrate present in plant and animal. Okay, so plant is which one? Which is the answer? What is the answer, please? Can I have the answer? Tak nak lah, saya je yang bagi.
C kot.
C.
E kot.
Urine. Mana awak jumpa urine ni?
Bakteria.
Bakteria dekat mana bakteria punya bahagian?
Cell wall. Dunia cell wall.
So bakteria ni animal ke?
Plant.
Plant? Animal? Bakteria plant?
Tak tahu.
Dah belajar chapter 2 kan? Maybe saya nak kena buka kamera saya supaya awak rasa takut sikit. Bacteria falls under which category? Is bacteria
Prokaryote.
Prokaryote.
Kan, so organism, okay, you can divide organism based on the type of cell, kan. So it either can be prokaryote or eukaryote.
Eukaryote.
So kalau eukaryotes, you have plants and animals. So basically ini ditanyakan pasal eukaryotes, okay. So obviously B and C salah lah kan. So kalau urine bukan dalam bakteria juga kan. So the answer will be A lah. Okay, so plant, the storage carbohydrate will be starch. Okay, so starch are found in rice, Okay, in plant lah, in rice grain, okay, in potato tubers, and then you can also find starch in legumes, ke kacang, okay. And then for chitin, it is the component of exoskeleton in of insects. Okay, so insect, a grasshopper and all that can. So you can find chitin in the component of exoskeleton of insects or arthropods. Okay. So number 20, which of the following contributes to long-term energy storage in animals? So starch salah, cellulose salah, triglyceride salah. Okay, salah. Sekejap. Jawapan dia adalah long-term storage in animal. Long-term energy storage.
Lipid.
Kenapa jawapan dia bukan lipid lah? Patutnya boleh jadi triglyceride kan? Okay, tak apa. Jawapan kat saya ni dia bagi glycogen. Tapi glycogen not as long as kalau triglyceride. If your body kan is lacking glycogen kan, kalau badan awak dia akan pergi kepada glukos dulu kan. So glukos as instant fuel to carry out cellular respiration. Okay. So kalau dah tak ada glukos dalam darah, so dia akan break down glycogen which is stored in the liver. Okay. So kalau dah tak ada glycogen dah kan, so awak dah tak makan, dah patah hati semua, dah tak nak apa-apa. Okay, contohnya lah kan. So badan awak akan pergi ke ataupun awak mengamalkan diet yang Atkins whatever. So dah tak ada glukos, dah tak ada carbohydrate, so dia akan pergi kepada lipid, triglyceride. Okay. So jawapan ni supposed to be D lah. Saya tengok balik. Saya tanya cara lain. Tapi saya rasa jawapan dia D. Okay. Okay. Number 21. Which of the following functional group is not found in fatty acids? Jadi dah masuk soalan fatty acid, dah masuk soalan lemak ni kan. So tengok dia punya flow dia. Nampak Kalau inilah flow dalam soalan exam, kalau macam contohnya kalau dalam macromolecules kan, so dia akan tanya pasal yang first chapter, okay, first content of the chapter, so dia akan tanya pasal bonds kan. So kalau tengoklah kat sini kan, so dia akan tanya pasal water, alright. So tengok flow dia. Then so lepas tu dia akan tanya pasal protein, ikut nota turutan lah kan. Lepas tu ni sekarang tanya pasal carbohydrate, and then lipid, and then dia akan tanya pasal nucleic acids. Itu turutan dia lah. Kalau dekat asasi ni, you never ever spot soalan. Ini bukan SPM so you never ever try to spot soalan because all the content which is the question dah disetkan untuk semua bahagian topik tu, subtopik tu ada tendensi untuk dikeluarkan untuk ditanya dalam exam. Ya, dalam exam ke dalam test. In this case chapter 1, chapter 2 dia akan ditanya masa test. Tapi kalau untuk final exam dia tak tak ditanya dalam final exam. Final exam chapter 3 sampai chapter 8. Okay, chapter 1 and 2 will be asked for your test lah. Okay, in your test. Okay, so okay, back to the question. Which of the following functional group is not found in fatty acid? Okay, so which one? Ada hydroxyl, ada carbonyl, ada carbonyl ke?
Ada
Ni ada.
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 Sida berapa?
Sila berbond O dah ada. And then phosphate. Sekejap Carbonyl ke? Itu bila dah jadi triglyceride kan? Fatty acid, in the structure of fatty acid, it has carboxyl, C, double bond, O, OH. So when the fatty acid is going to link to glycerol, then condensation reaction occurs, okay, which remove OH, which removes OH to link to the glycerol, So then baru akan jadi triglyceride kan. So maybe dalam dalam structure triglyceride, then you have the carbonyl. Okay, tapi kalau fatty acid, it does not have yet the carbonyl group. Tapi maybe they consider, okay, tak apa. Tapi yang paling tak ada phosphate. Okay, yang paling tak ada phosphate. Kalau dalam fatty acid, it has the carboxyl. Bila dia jadi triglyceride, then only dia akan jadi daripada carboxyl dia jadi carbonyl. Okay. Tapi yang paling tak ada kena-mengena dengan fatty acid ataupun triglyceride is phosphate. Okay, so tak apa, ini saya tanya dekat orang pensyarah lain. Okay, so unsaturated fat. Lecturer awak siapa? Madam Liza ya?
Madam Aida.
Beda Aida.
Nurul Aida.
Okay. Maybe awak boleh tanya ke Madam Aida juga soalan 20-21 ni. Nanti saya tanya pensyarah lain juga. Okay. Okay, so number 22. Unsaturated fat is a straight molecule, consists of 6 double bond, is liquid at room temperature, have maximum number of hydrogen bond. So number 4 ni salah lah kan. So kalau maximum is supposed to be saturated fats. Unsaturated due to the presence of what? Double bond kan between the carbon kan. So when you have double bond between the carbon, so the carbon is bonded only to one hydrogen, each carbon that has the double bond. So when you have the double bond, it causes the molecule of the fatty acid to be bent. That it is bent. So when the molecule is bent, it has kink kan. So this prevents the fat molecule from packing close together at room temperature kan. So dia tak boleh nak rapat-rapat antara satu sama lain at room temperature, so dia dalam keadaan liquid. But as for saturated fat, okay, because the molecule is straight, they don't have double bond kan. So at room temperature they can closely pack together, so they are solid at room temperature. So this is for saturated fats. Tapi in this case, it is unsaturated fat. It is liquid at room temperature. So yang ini betul lah. So nombor 3 betul. Okay. So now we are left with
B.
B. Okay. So consists of 6 double bond. So ini betul lah. Okay, is a straight molecule? Salah. Straight molecule is for saturated fat due to the presence of saturated fatty acid. Awak kena take note of these two term unsaturated fat, komponen dia adalah unsaturated fatty acid. Komponen dia adalah unsaturated fatty acid and also glycerol. Faham Okay. So istilah dia awak kena take note. Unsaturated fat is the fat itself, minyak sayuran kan, whatever, minyak ikan. So that is unsaturated fat. The component is glycerol with the unsaturated fatty acid linked together through the formation of ester linkage. Okay. So kalau sama juga kalau saturated fats such as animal fat, butter, lard, okay. So the component is glycerol, which is bonded to saturated fatty acid. Okay, so please take note of this. Okay. Istilah tu sangat penting untuk awak take note. Fat and fatty acid is different. Fat is the fat, the component of fat is fatty acid. Okay. So number 23, which of the following are the components of a nucleotide? So obviously dia ada nucleotide ni, it has 3 component lah, which is phosphate bonded to 5 carbon sugar. It can either be ribose or deoxyribose, depend on the molecule. If it's DNA, it is deoxyribose. If it's RNA, it is ribose. Okay, and then the ribose is bonded to a base. Okay, so yang ini salah sebab tak ada apa, tak ada base. Ini adenine and deoxyribose salah sebab apa, tak ada phosphate. Okay, sugar nitrogenous base salah sebab apa, sebab tak ada phosphate. Okay, and then the answer will be D lah. Okay, so adenine is the nitrogenous base, ribose and then phosphate. So this molecule is basically the nucleotide for which molecule? For which polynucleotide? Which type of polynucleotide? This one. D ni. Polynucleotide, nucleic acid, you have DNA and also RNA. Okay. Apa yang membezakan DNA and RNA is the 5 carbon sugar. So for D, which one is it? Is it DNA or is it RNA?
RNA.
RNA. Okay, so RNA. So another difference between the structure of DNA and RNA is the base. Okay, is the base. Kalau base untuk RNA, you have the adenine, guanine, cytosine and uracil. But in DNA, you have adenine, guanine, cytosine and thymine. Okay, so you have no thymine in RNA. Instead, you have uracil. Okay, so DNA you have thymine, don't have any uracil. Okay, so that one you have to take note lah. Okay, so number 24, in nucleic acid, 5 carbon sugar are linked by a bonded to a phosphate by a okay, so you have to name the bond. Sekejap. So what is your answer for 24 ni?
Phosphor.
Phosphor?
Diester.
That is the linkage or bond. Okay. So phosphodiester bond or phosphodiester linkage is the bond that links together the nucleotides. Okay, between the nucleotides. Okay, the nucleotides monomer within DNA or RNA. So kalau you look at this, okay. So this is the basic structure. So this can be adenine, okay, and then the 5 carbon sugar is linked to a phosphate group. Okay, so this is the basic structure of a nucleotide. So this is ini oxygen kan? So carbon number 1, number 2, number 3, number 4, number 5. So number 3 it has OH. Okay, it has OH group. So that's why kalau tengok dalam in the labeling of DNA strand kan, so dia ada DNA strand, the double bond, right? The double strand DNA. It is helical. Tapi ni saya lukis dia straight supaya awak faham. So dia ada directionality dia. Okay. So the top strand you label it, it can be labeled as 5 prime and the one here is 3 prime. So obviously kalau 5 kena complement dengan 3, kalau 3 kena complement dengan 5. Okay. So along the strand, if the bases are adenine, okay, the other strand has to be thymine. Okay, thymine. Guanine, cytosine. Contohnya kan, cytosine, guanine, thymine, adenine and all the way through lah. Okay, so kenapa dia 5 prime? Okay, so 5, kalau strand tu start dengan 5 prime, so the direction adalah daripada 5 ke 3. Okay, so dia akan pergi in this way. Okay, 5 ke 3. So kalau bottom strand is 5 to 3 prime. So yang ini kita namakan dia sebagai anti-parallel. Okay, anti-parallel. Okay, sebab 5 ke 3, bawah pun 5 ke 3. Alright, itu cara awak baca dia. Kenapa arrow dia ke 3? Sebab apa? Every time, okay, when you want to add more nucleotide, okay, to the DNA strand, to each of the DNA strand, the new nucleotide is always added at the 3 prime end. Why? Kalau tengok kat structure ni, kan, you always add new nucleotide at carbon number 3. Kan? So carbon number 3, you keep on adding new nucleotide at carbon number 3 because at carbon number 3 you have the OH, you have the hydroxyl group. Okay? So kita belajar kan, in order for you to link together the monomers, tak kisahlah ada glucose ke, untuk jadi untuk jadi longer polysaccharide such as starch kan, ataupun it is amino acid, you want to join together all the amino acid to form polypeptide, okay, or you want to join together nucleotide, okay, to form polynucleotide in nucleic acid. So all the reaction where you join together all the monomers is through the reaction of condensation ataupun dehydration. So the reaction involves the removal of water molecule. So in this case, at carbon number 3, they are the OH. So so you want to add the next what you call it, the next nucleotide. Okay, so the next nucleotide will be added where, saya padam sekejap why, okay. So awak kena tahu kenapa strand DNA ni ataupun RNA we label them as 5 to 3 prime. Okay, because every time when you want to add new nucleotide to the strand, they are always added at the 3 prime end. So for example, okay, so this is the next nucleotide to be added. Okay, so ini let's say lah guanine ada kat sini, phosphate. Okay, ini saya padam dulu. Okay, phosphate. Okay, so phosphate. So in the process of adding the next nucleotide, this is carbon number 3 ada OH kan. Alright, so in the process, okay, phosphate awak kena tahu structure dia macam mana kan. Phosphate PO4 2 negative kan. Sebab apa negative? Initially the phosphate ada 4 oksigen, right? Okay, OH kan, macam tu kan. So initially yang mana ada O negatif ni asalnya adalah dalam keadaan OH kan. Okay, so in the process of condensation ataupun in the process this molecule are in aqueous condition, so they are ionized kan. So ionized dia akan jadi O negative. So initially ada H kat sini. So in the reaction of attaching the phosphate to the previous nucleotide in the strand, so dia akan reaction tu akan menyebabkan OH remove with the hydrogen ion, okay, so to form water. Okay, so now OH is removed, so the phosphate is now linked. Saya buat dengan one biru lah kan. It's now linked, okay, together. So this is carbon number 5. So the carbon number 5 ni dia tak terlibat dalam reaction. Yang terlibat dalam reaction to join the nucleotides together is carbon number 5 and the phosphate of the next nucleotide to be added. Okay, to be added. Okay, so itulah dia. So itu awak kena faham kenapa dia 5 to 3 prime. So 5 prime, 3 prime. So every time you want to add new nucleotide, it's always added at the 3 prime and sama je applied kat DNA and also RNA. Okay, so number 25, choose the correct component of a deoxyribonucleotide. Okay, so deoxyribonucleotide is DNA lah, kan? Nucleotide purines. Dalam nucleotide memang dah ada purine. Memang dah ada purine. These are bases kan? Base. Okay, so check balik. Dalam purine apa base dia apa? Okay. Nucleoside, adenine. Nucleoside is without the phosphate, right?
Yes.
So nucleoside ni dah ada base dah dalam dia. So salah. Sekejap saya check. Okay. So kalau dalam DNA tak ada uracil, so salah. So betul lah yang so it leaves you with D. Okay. So phosphate link, okay. Link phosphate link to 5 carbon sugar, okay, with thymine. So the answer will be D lah. Okay. So ingat ya, nucleotide salah sebab apa? Dalam nucleotide memang dah ada purine. So memang dah ada base kan. So purine is a base. So dia takkan ada dua base link together. So one nucleotide will only have one base. Nucleoside and adenine salah sebab apa? Tak ada phosphate in this molecule. Okay, lepas tu dah ada dalam nucleoside dah ada base. So salah lagi. Macam itulah, kena faham kenapa dia salah. Okay. So number 26, which of the following statement describe the structure of nucleic acid? Okay, which of the following statement describe the structure of nucleic acid? So kita tengok dia punya statement dia. Okay, so number 1, the backbone is positively charged. Betul salah? Nucleic acid, is it positively charged or negatively charged?
Negatively charged.
Negatively charged. So salah. Kenapa negatively charged? Tadi saya cakap sebab apa? Due to the presence of phosphate when it is in aqueous solution, in aqueous kan, environment in your cell, and your cell is aqueous, okay? So dia akan ionize. So dia akan jadi PO4 to negative in the molecule. So that's why molecule DNA, RNA ni we call it as acid. Okay, ribonucleic acid, deoxyribonucleic acid. So they are acid due to the presence of phosphate that has negative charge. Okay. So number 2, a phosphate group link the sugar of 2 nucleotides. The phosphate group that link the sugar of 2 nucleotides. Betul lah kan? So the phosphate group that link the sugar. Okay, that link the sugar between the 2 nucleotides here. So number 2 statement, is it wrong or not? Is it correct or not?
Yes.
No.
Correct.
Alright. So adjacent nucleotides are joined by a phosphodiester linkage. Betul lah kan? So the bond here kan. So ini is the phosphodiester linkage. You have to know how to, kena tahu tak kat mana bond tu So ini adalah bond dia. Okay, bond dia. Okay. Between the carbon of carbon number 3 of the previous nucleotide and carbon number 5 of the next. So dia dalam tu ada phosphate. Saya check. Kena tahu macam mana nak label phosphodiester bond. Dalam exam dulu kalau tengok soalan-soalan past year ke whatever kan. So you have to know how to, where to label. So kalau nak label kan dia, jangan circle the 2 carbon So kalau circle terlebih, you include in the bond the carbon, your answer would be wrong. Kalau terlebih sikit kan, salah. So the bond does not include the carbon. does not include the you circling the carbon. So it is macam ni je awak labelkan dia. Macam tu. Okay, macam tu. So that is the phosphodiester bond. Okay. Sebab bila awak circle carbon tu, dia akan dah jangkau ikatan covalent dalam carbon kat sini. So dia akan jadi salah lah. Okay, so please tengok balik awak punya nota. Takat mana dia labelkan phosphodiester bond tu. Okay. Okay, so number 3 betul. Nucleoside without the phosphate group is called nucleotide. Betul salah ni?
Salah.
Sebab dia terbalik.
Terbalik.
Okay, dia terbalik. This one supposed to be nucleotide, bawah ni baru nucleoside. Okay, so the answer will be 2 and 3. Okay. Okay, so do you guys have a class after this? Kalau tak ada, saya nak sampaikan sampai pukul 9.
Tak ada.
Ada kelas pukul 10.
Okey.
Okay, so kita continue. Okay, alright. So part B, structure question. Okay, for question number 1. Okay, it says it shows you here. Okay, figure 5 shows 2 types of nucleic acid, which is you have DNA and also RNA. Okay. Okay, so name the monomer of nucleic acid. Alah, senang je ni. Apa dia? Ni markah free je. Soalan dalam exam ni
Nucleotide.
nak bagi markah free. Okay, nucleotide. Okay. Okay, so okay, ini awak kena kira-kira sikit. Okay. So if 15% of the bases in a particular DNA molecule is thymine, okay. 15% is thymine. Okay. What is the percentage of cytosine? Okay, in the same molecule. Show your calculation. Boleh tak bagi saya an instant answer? Saya nak tahu apa jawapan awak. How do you do this calculation? Can anyone give me an instant answer so that I know, oh, okay, adalah yang faham soalan ni. Cuba saya bagi masa seminit 2, cuba kira.
Madam,
Ya?
jawapan dia 45% ke? 35% ke?
Jawapan dia 35%. Siapa jawab tadi? Saya tak perasan nama.
Nurul Ain.
Okay. So how do you do your calculation, Ain?
Thymine 15%, dia akan pasangan dia A. So A tu akan 15% juga. Jadi 100%. Jadi jumlah dua tu 30%. 100% tolak 30% dapat 70. 70 tu C dan G.
Adalah apa? Comprises of guanine and
sitosin.
So ini, so maksudnya kena halfkan je lah 70 tu kan. So each guanine and cytosine ni adalah 35%. Faham yang lain?
Faham?
Alright. Okay. So thank you Ain. So it shows that you understand. So okay lah. Okay, bagus. So soalan ni awak kena tahulah kan, complementary base pairing tu adenine kena pair with thymine, guanine kena pair with cytosine. Okay. So all these okay adalah dalam molekul DNA ke kan ataupun RNA ke, so all together kan, so mestilah 100% of bases. So kalau dia dah bagi a certain percentage kan, daripada situ awak boleh dah kira kan. So kalau adenin dia punya amount mesti sama dengan thymine, guanine amount dia mesti sama dengan cytosine. Okay, so kalau dalam lagi satu saya nak emphasize, okay, dalam DNA strand kan, so the adenine pair with thymine, guanine pair with cytosine, okay, so the bond between the bases is hydrogen bond. So ingat kalau adenine and thymine, the bond, there are 2 hydrogen bonds, okay, that hold the adenine and thymine together. Kalau guanine and cytosine ada 3 hydrogen bond, okay. 2 hydrogen bond antara adenine and thymine, 3 hydrogen bond between guanine and cytosine. So itu saya nak emphasize jugalah. Okay. So you have to know. Kalau dalam exam dia suruh lukis hydrogen bond, so remember always draw hydrogen bond using dotted line. If you draw using straight line, so dia dah jadi salah. So sebab tu that will indicate that the bond is a type of covalent bond. Okay. So number 3, DNA is double stranded while RNA is single stranded. Give another structural difference between DNA and RNA. Okay, take note kalau soalan tu suruh bagi perbezaan, in this case perbezaan yang spesifik yang dia nak adalah structure. If for example kan carelessly tak baca dia punya keyword ni, tolong highlight keyword dia nanti, structural difference. If for example okay, DNA are found in the nucleus, dah confident dah siapa awak ni kan, in the nucleus. RNA are found in the cytoplasm and also nucleus. Dah betul dah, nampak macam betul salah. Sebab apa? Dia nak structural difference. Okay, structural difference. So in this case, one of the answer can be in terms of the ribose sugar. Okay, the 5 carbon sugar. So the 5 carbon sugar in RNA is ribose. Awak boleh bagi tahu kan. So the 5 carbon sugar present in the nucleotide of RNA is ribose. For DNA, the 5 carbon sugar that are present in the nucleotide of RNA is deoxyribose. Itu perbezaan yang pertama. So perbezaan dia mesti apple to apple mesti apple to apple. So baru awak boleh dapat 1 markah. Okay, kalau let's say lah awak bagi perbezaan dia buah apple dengan buah durian, salah lah kan. Contohnya awak bagi okay, dah betul dah fact, okay, the 5 carbon sugar in DNA is deoxyribose, tapi tiba-tiba sebelah ni awak bagi tahu the bases that are found in RNA is adenine, guanine, cytosine, uracil. Facts dah betul dah, tapi you are not comparing benda yang sama. Okay, so facts dah betul tapi you are not comparing apple to apple, so salah kan. So kena benda yang compare tu benda yang sama. Okay, so baru betul. Okay, so in this case kalau nak bagi tahu in terms of bases, bolehlah juga kan. So kalau kalau RNA, the bases are guanine, adenine, cytosine, uracil. Kalau sebelah sini adalah adenine, thymine, guanine and cytosine. Okay, so itulah that are the differences. Okay, the differences between the molecule. And then kalau bagi tahu in terms of structure juga, kalau DNA the molecule is larger. Kalau RNA the molecule is smaller kan. So itu boleh one of the answers that can be include lah. Okay, ataupun for the 5 carbon sugar at carbon number 2 of deoxyribose ada OH. Ada H. Okay, kalau at carbon number 2 of the 5 carbon sugar you have the hydrogen. Okay, so ada berapa dah saya bagi? A few lah kan. Okay, ada a few yang awak boleh take note. So dia nak 1 markah je. Okay, so it is now 9 o'clock. Kita stop dulu sampai sini kelas. Okay, ada soalan tak?
Saya ada. Kalau yang soalan yang difference tadi tu, RNA dengan DNA tu, so kita sebut je RNA tu have uracil, DNA have thymine. Boleh ke? Macam tak sebut semua.
Boleh lah. Sebab yang adenine, guanine, cytosine ni persamaan lah, kira persamaan dia lah kan. Okay. Dia nak perbezaan. So boleh lah, okay je.
Okay, madam.
Okay, ada soalan lain? So kalau tak ada soalan lain, kita stop dulu kelas. Attendance tak apalah kot, saya ada record dia. Okay? Alright. Tak ada So kita end dulu sampai sini. Thank you for your attention. I'll see you next week. Okay?
Okay.
Thank you, Miss.
Thank you, madam.
Thank you, Miss.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Bye.
Thank you, madam.
Thank you, madam.
Alright.
Thank you, madam.
Thank you, madam.
Okay.